Hi, my name is Emma Atkinson, and I go to Northwestern High School. I did my science fair project based on Catalase Activity. So my question was, does the concentration of hydrogen peroxide affect the activity of catalase? My hypothesis was, if there was stronger hydrogen peroxide added to beef liver, there will be more activity resulting in more bubbles. The weaker the hydrogen peroxide, the less the activity. The purpose of this experiment was to determine if the strength of hydrogen peroxide played a role in the activity of catalase. In this experiment, the size of the balloon determined the activity of the liver. To read more about the purpose of this experiment, you can go to my abstract in my paper. So before you can do a catalase activity, you have to know what catalase is. So catalase is a very common enzyme present in almost all organisms exposed to oxygen. This enzyme is found in the liver, helps break down peroxide into water and oxygen. When catalase is added to hydrogen peroxide, there's an initial rapid evolution of oxygen, which lasts for about 2 minutes. The activity site is where the bubbles and fizzing, or the reaction, is taking place. The normal concentration of hydrogen peroxide is 3%. Any other concentration can cause results to vary. Materials and procedures. So materials needed were beef liver, 3 beakers, a knife, graduated cylinders, scale, 15 test tubes, a tray, balloons. I recommend more than 15 because they break very easily. Hydrogen peroxide of all concentrations, 1.5, 3, and 6, a dropper for liquid, a ruler, gloves, a cutting board, and wax paper squares, and some sort of data collection, and I use Google Sheets. So procedures, I took the liver slice and I cut it into chunks. I measured them so that they were all 2 grams. I put them on a scale and made sure that they all measured to be 2 grams and the same. Then I put them into the test tubes. I swirled them so that they would sink to the bottom. Once the liver was in the bottom, I took the test tubes and placed them so that they were in sections of 5, so that there were 3 sections, and then I put 1.5% hydrogen peroxide onto them, and then I placed the balloon on as fast as I could, and then I repeated that with every other test tube. For 5 of them, I did 1.5, for another 5, I did 3, and for another 5, I did 6. Once I was done, I measured the balloon diameter and I placed that into my sheets so that I could collect that data. Then I cleaned up all materials used. These are my results, and this is just an average. As you can tell, I didn't put all the results in, but this is the basic idea. And basically, the lower the hydrogen peroxide concentration, the lower the activity, and the smaller the balloon diameter. So conclusion, shown by the results in this catalase experiment, the stronger the hydrogen peroxide concentration is, the more activity and bubbles produced. This is another graph showing the results, and these are my pictures. Discussion, looking through the results, the higher the concentration of the hydrogen peroxide, the fuller the balloon got. The highest or strongest percentage, 6%, resulted in the most filled balloons. Within the background research, it talks about the activity site and the reaction itself. While doing this experiment, that was proven to be right. Once the hydrogen peroxide was added, there was instant pressure in the balloon because of the enzyme activity. So discussion continued. You can also find this in my paper. And these are my cited sources. Thank you so much for listening, and have a great day.